Okay, so can you able to see the screen? Okay, can you able to see the screen? Okay. Uh, Asya Shaikh, what happened? Asya? Okay, okay. No, you raised your hand means you have any doubt you can ask. Okay. So, in drug design, there are two important types of methods are there. One is structure-based drug design. Another one is ligand-based drug design. Okay. So, the drug design may be any types, but for that purpose, what we need to have a lead compound or a drug compound whose structure is known, and a target compound, target protein whose structure known or maybe not known. So before going to do any kind of drug design, first we need to have the database of the drug molecules and the database of the proteins or target biological compounds. Okay, so in order to know their structures, okay, there are different kinds of techniques generally they use. They may be NMR technique or they may be X-ray crystallography technique. Okay, so based on these techniques, okay, all these drugs and their structures, their properties, okay, even the structures of proteins and their properties were identified. Okay, for example, if you see in the slide here, in X-ray crystallography, okay, by following the X-ray crystals on the surface, on the crystal or on the drug moiety, okay, it creates an electron density map around the skeleton of the drug. So based on that, a model were drawn. Okay, an atomic model of the protein or the drug were drawn. So by using these X-ray crystallography techniques, okay, we usually know the structures of chemical molecules or protein molecules or any target molecule. So like that here, based on the structures which are identified by different kinds of biophysical processes or biophysical techniques such as NMR and X-ray crystallography techniques, various kinds of three-dimensional structures are known. Okay, all these structures are compared with each other and different kinds of models were formed. These models are called homology. This is called homology modeling. Okay, so all these models and their structures were formed in a kind of database. For that proteins, it is called protein database. If you want to know any kind of protein on which we need to apply our drug molecule, we have to go to that protein database. Whatever the name of the protein, we have to type it them in that, and the protein structure will come because the database is already identified based on these techniques. Okay, so based on the structures of drugs and target molecules, if we do the drug designing, it comes under structure based drug designing. So this structure-based drug design is one of the several methods in rational drug design toolbox. So these drug compounds are designed to inhibit, to restore, or otherwise modify the structure and behavior of disease-related proteins and enzymes. Okay, so the based on the behavior of these proteins and enzymes and their structures, we need to modify the structures of drug compounds, and based on that we need to design the drugs. Okay, so this comes under structure based drug design, which is also called as SBDD. So SBDD uses the known 3D geometrical shape or structure of proteins to assist in the development of new drug compounds. So based on the geometrical shape, 3D geometrical shape, or uh, 3D geometrical arrangement of proteins and enzymes, based on that new drug compounds are developed. Okay, uh, all of you listen here. From this time onwards, different types of terminology come into the picture, and there is explanation is there, there is simple elaboration also, both mentioned in the slides. Okay, so meanwhile, if you are getting difficult to understand any sentence or any line or any explanation, please ask, because from this time onwards some complexity and at the same time some simplicity will be there in the explanation. Okay, whenever I explain some topic, in a complexity, in next slide I'll try to simplify them. Okay, but whatever it there, you need to understand whatever I'm trying to explain. If you are unable to understand, okay, you can message me or you can ask me immediately so I can simplify it. Okay, so based on these 3D geometrical shapes, which are generally identified as protein targets or target moieties, okay, their structure were often derived from using X-ray crystallography or NMR techniques. So both these X-ray and NMR methods can resolve the structure of proteins to a resolution of few angstroms. So we can able to increase the size, or we can able to go to a particular fragment that protein, we can observe their structure. So like that here, both X-ray crystallography and NMR made it possible to identify different kinds of three-dimensional structures of our target proteins. So it made easy in order to design a new drug. So at this level of resolution, researchers can precisely examine the interactions between atoms in protein targets and atoms in potential drug compounds that bind to the proteins. Okay, this ability to work at high resolution with both proteins and drug compounds makes structure-based drug design one of the most powerful methods in drug design. So the beauty of the SBDD, the beauty of this structural based drug design method is extremely high level of detail that it reveals about how drug compounds and their protein targets are interact, because we have a three-dimensional approach is there in front of us, so we can able to see the protein, how their structures are there. Okay, so based on that we can easily understand how our drug is able to interact the protein target which is already there in the database. Okay, so there are certain important points we need to understand in structure based drug design. Structure based design is one of the first techniques to be used in drug design. So if you are going to design a new drug, the first technique is we have to follow is the structure based drug design. So based on the structures of both drug and protein, we have to start our designing technique. Structure based drug design that has helped in the discovery process of new drugs. In parallel, information about the structural dynamics and electronic properties about ligands are obtained from calculations. So, based on this structure based drug design, okay, we'll get the information of those structural dynamics and their electronic properties of the ligands. So there are computer based calculations will be there automatically. They will give you the information. So this has encouraged the rapid development of the structure based drug design. And this structure based drug design can be divided roughly into two categories. Okay, it is either ligand based drug design or database searching. Second one is receptor based drug design. So both again comes under SBDD only. Okay, first one is ligand based drug design. Second one is receptor based drug design. So generally, the NMR spectrometer and X-ray crystallography helps us to identify the structures of different kinds of drug designing. Uh, it is here. So if you see the general methodology of the structure-based drug design, first 3D structures of the target molecules is known, which is also called as homology modeling. So from this, we can able to generate the 3D structures of target molecules, okay, which is closely related to the structure of proteins. So from that, we have to detect active sites. What is the site of which is the active site where the drug will bind? So we have to first detect the active sites. Based on the active sites, we have to dock our target compounds from the database. So like that here, different kinds of ligand structures and their list are determined, and from that, based on the efficacy, all these things, the different types of ligand structures are refined, and each of the ligand structures is analyzed to find the sites in the ligand involved in binding to the targets. So from a list of ligands which are refined, from that, which kind of ligands are able to bind the active sites of the targets were identified, and in next, those sites on the ligands can be optimized to increase the potency uh, involved in the designing of the ligand. And these ligands next have been designed and then synthesized in the laboratory, and they're tested through the cell-based or biochemical assay. Means further enter into the clinical trial phase. Once the clinical trial is done, the commercial drug came into the picture. All these things start from the structure-based drug design. But if you face any difficulty in order to understand this, okay, let me simplify it. Okay, so SBDD means structure-based drug design is of generally two types. One is ligand-based drug design. Another one is receptor-based drug design. Okay, in ligand-based drug design, the first category is finding the ligands for a given receptor, which is usually referred as database searching. Okay, means finding the ligands for proper receptor in the database is the first step in the ligand based drug design. Once we search database and find out the proper category of ligands and receptors, then we have to go for next step. In this case, a large number of potential ligand molecules are screened to find those fitting the binding pocket of the receptor. Binding pocket is nothing but the active site present on the receptor is identified based on which kind of ligand is properly fitting in that active site. So, like that, based on that, various kinds of ligands were screened. So the list of ligands will automatically come into the database. This method is usually referred as ligand-based drug design. And the key advantage of database searching is that it saves synthetic effort to obtain new lead compounds. Okay. So, for example, new disease came into the picture, and based on that protein which is responsible for the disease in that virus or any microorganisms, we uh, based on the literature search, we first identify few drug molecules and we go for synthesizing. But it takes lot amount of time. If we do this entire thing, we have to first literature search. We have to do from that literature database, we have to uh, find out few molecules and we have to go synthesize, then we have to test in clinical trials. It takes lot amount of the time, right? So in order to save the synthetic effort, first we have to go for the database searching, based on computers. Then it will give a list of lead compounds, list of lead structure drug molecules, which can able to efficiently fit in the active sites of proteins or target molecules. Based on that ligand structures, we can directly go for synthesis, right? So ligand based drug design saves the synthetic effort of the scientist in the laboratories, based on the database which gives them the list of ligand compounds. So this is briefly the ligand based drug design. Receptor based drug design is based on the structure of receptors, means the proteins or other target enzymes or other things on which the ligand should bind. So this is another category of structure based drug design method, which is about building ligands, which is usually referred as receptor based drug design. In this case, ligand molecules are built up within the constraints of the binding pocket by assembling small pieces in a stepwise manner. Okay, so ligand molecules are built in a stepwise manner based on the difficulties arised when these ligands are binding to the pocket or active site of the receptors. So, so like in a small pieces, piecewise, the ligand molecules they started building based on the active sites of the receptors here. Once these pieces can be either individual atoms or molecular fragments, when they properly arranged or properly binded, then based on that, the receptor drug design will be done. The key advantage of such method is that novel structures not contained in any database can be suggested. Means new kind of structures of receptors and new kind of structures of ligands were identified and a new database is created. Okay, so this comes under receptor based drug design. Okay, so if we try to understand as a flowchart of structure based drug design, okay. So in this structure based drug design, first we have to go for in silico kind of drug design. In silico means in a database, in a computer. If we do all these kinds of drug designing, it comes under in silico kind of activities. Once we find a lead compound, then we have to synthesize and we have to analyze and we have to test on a microorganism or on a patient. It comes into in vitro kind of activity. Okay, so before doing in vitro kind of test, we have to do in silico activities. All this comes under computer aided drug designing. Okay, so molecular modeling, computer based drug design comes under in silico kind of activities. So this structural based drug design is an important bridge kind of thing which helps for both in silico and in vitro kind of activities. Okay, so in preclinical stage, first we have to design our uh, ligands, and based on the structures of these lead molecules which are able to efficiently bind to the lead molecules, the computer database will give the rankings to those molecules. Okay, based on those rankings we will decide the lead molecules which will show their efficiency in the active binding to the target molecules. So from this lead molecule, few lead molecules we try to synthesize in the laboratory. Once we synthesize these chemical molecules, they will be analyzed for their structures, their binding activities, their properties, their chemical properties, all the properties of those molecules were analyzed, and again it enter into the preclinical stage where they will be tested. Again from this, again one or two compounds are finally identified which enter into the clinical trial mode. So like this, all these activities, all these steps will be done based on the structural based drug design only. Okay, so these structural based drug design generally based on pharmacophore or the receptor here, are based on the ligand here. Okay, so first in receptor based drug design, the pharmacophore of the receptor will be identified, and in next step the pharmacophore will be modified. If the receptor is fit, we will go for potential drug design, or it is not fit, again we have to go back for the pharmacophore modification. So like that, the receptor based drug design flowchart will be look like this. Okay, in ligand based drug design, first active site of the receptor will be identified, and that the ligand fragments will be docked or applied. Okay, if the ligand is properly fixing or fit for the receptor, okay, we will go for complete growing of the receptor and ligand moiety. Okay, if it is completely fixed, we will go for potential drug synthesis. If it is not fit, again we have to modify the receptor and ligand Okay, we have to change the fragments of the ligands. Again, we have to try to fit that ligand into the uh, receptor. When it is again properly fixed, properly bind, okay, the drug will be identified as a potential drug. So like that, it is stepwise manner, like a flow chart, where we have to follow different kind of steps in the structure based drug design. Okay, so the first type of structure based drug design is the ligand based drug design, okay, which is very important one. Okay, in ligand based drug design, ligands came into the picture, okay, which plays important role in the drug designing. So, general methodology of structure based drug designing begins from identifying the 3D structures of target molecules. Okay, these 3D structures generally available in the structural database. Okay, let me show the database. Okay, I'm sharing another tab here. Okay, so this is protein data bank. Okay, you can also able to visit it in online. Okay, it is called rscb.org, which is called protein data banks. So there are various kinds of proteins are deposited, means their structures are deposited based on the research activities which are publicized or reported worldwide. Okay, so here we can find out different kinds of proteins online and their structure. Okay, so for example, you are synthesizing an anti-TB drug, anti-tuberculosis drug. So before synthesizing that molecule, first you need to know which kind of protein in the tuberculosis causing bacteria functioning majorly. Okay, so if we are able to inhibit that protein, okay, whatever the drug molecule is able to cure the disease. So first we have to know the structure of that protein. For example, H37RV. 37RV is the common drug for common protein which is responsible for the TB tuberculosis activity. Okay, so if you search it, okay, it will give you the structure of H37RV. Okay, so here 3D view. The crystal structure obtained here means the crystal structure obtained by the X-ray crystallography technique. Okay, so this is the structure of that protein. H37RV protein looks like this. Okay, so from the protein data bank, we can able to identify the structure of these proteins. Okay, here all the properties of this crystal structure of protein is present here. Its measurements, its components. Okay, so different kinds of presets you can find here. Okay, how water is arranged around this protein moiety. Also, you can able to observe here. Okay, so you can go and search in different kinds of models also available here. Okay, 3D models are available. Okay, so if you zoom it here, okay, these are the various kinds of amino acid chains present in the protein structure. Okay, so like this here, the structure of the proteins were generally collected from the protein data bank. Okay, you can able to view in different kinds of uh, models, like different kinds of viewing techniques are also there. You can observe in molecular models, you can observe in uh, NGL model, you can observe in JavaScript model. So there are different types of options also there, you can able to find out here. Okay, so this is cartoon kind of structure, secondary structure. You can able to see different types of surfaces also there. Okay, so once we identify the proper protein database, see this is uh, in the presence of a solvent, how the structure of this protein will appear, look like this. All these are the different types of areas. So in which area this protein showing its activity, those are called active sites. Okay, in that we have to dock our protein, our ligand. Okay, so from this uh, structure we can able to identify what is the active sites are involving here, uh, by using by showing into different kinds of structure. Okay, in solvent accessible structure, in cartoon structure, in different kinds of models we can able to see, and we can identify which uh, protein is responsible for the different kinds of disease. So from protein data bank we can identify the proper protein structure, we can download it, and we can use it in our computer, where our target chemical moiety is able to dock it, means bind it. Okay. Okay, so once we find out the 3D structures of the target molecules, okay, and then we have to dock those protein molecules against our ligands in the database. Okay, so the ligated structures with best scores are then determined, refined, and analyzed to find the sites on the ligand involved in binding to the target. So the automatic scoring will be given in the computer software whenever our 3D structures of ligand are docked against the 3D structures of the protein moieties. So our target ligand molecules, if it able to bind properly on the active sites of the protein, so based on the binding rate, okay, the database itself give the ranking to the molecules, ranking to the ligands. Okay, again, these sites can further be optimized to increase the potency of the ligand, means which ligand efficiently binding to the target site, the potency will be more. More binding efficiency will be indicated by more potency of the ligand. So this is followed by cell-based or biochemical assay, followed by clinical trials. So based on this, the structure-based drug designing is identified as a proper process in order to identify both the ligands and target molecules. So based on that binding done by based on the database, we can able to uh, identify different kinds of ligand molecules. So that's why it is called ligand based drug design. If the potency of the ligands is poor, then next ligand in the list is tested through all the steps discussed above. Okay, so once the database give ranking to the ligands, one by one ligand is tested, how it is properly fixing. If it is properly fixing into the active site, its potency will be more. If it is not fixing properly into the active site, its potency will be poor. So like that, based on this, one by one ligand we have to test. So while choosing a drug target function, its prediction, its pathway information, and the disease association and structural data all must be, we have to look into it. We have to consider. Similarly, ligand selection must also be accurate so that it specifically binds to the target and produce a well-defined physiological effect, simultaneously minimizing the undesired side effects. Okay, so based on the rankings of the ligands and based on the binding energies or binding efficiencies from the database, we have to choose both ligand and target molecule properly based on their physiological effects, based on their minimizing side effects. All this will be you have to consider before choosing the ligands and their target molecules. Ligands used in drug designing must follow the Lipinski rules to be a successful drug candidate. Okay, so before choosing a ligand molecule, the ligand molecules must obey obey the Lipinski rules. Lipinski rules are there are four Lipinski rules are there. These Lipinski rules must be uh, followed or obeyed by the ligands. Then these ligands properly acts as a potential drug candidates or successful drug candidates. These Lipinski rules are five or fewer hydrogen bonds should be there. Okay, in the ligands, five or fewer hydrogen bond donors should be there. Second one is 10 or fewer hydrogen bond acceptors should be there. Molecular weight is less than or equal to 500 should be there, and calculated log p uh, less than or equal to 5. Okay, means log p is the uh, its acidity, or its a specific ratio. Okay, this log p value should be either equal to 5 or less than to 5. So these are the four important rule should be obeyed. Okay, so the ligand must contain 5 or fewer hydrogen bond donors. Second one is 10 or fewer hydrogen bond acceptors, and molecular weight should be less than or equal to 500, and its log p values will be less than or equal to 5. So these four Lipinski rules should be obeyed by the ligand molecules. Then it is considered as a successful drug candidate, and we have to choose it as a docking purpose. Okay, so this is where after choosing both ligands and target compounds, target receptors, then we have to do the docking. Okay, which is called docking of the ligands or molecular docking. Okay, which is very important. Molecular docking, it is the most common method which is widely used in structure based drug designing. Molecular docking is a method to characterize the behavior of small molecules and binding site of the target protein by modeling the interaction between a small molecule and protein. Okay, so molecular docking is a method where the small molecules of ligands and binding sites of the target protein should be modeled based on the interactions between them. How our target molecule, target protein is interacting with the protein. If it is not properly interacting, we have to model it again. Again, we have to dock it. Again, we have to dock it against each other, both target and ligand. Once the binding is happen properly, okay, our target interactions will be observed here. Okay, it is comes under molecular docking. So in this molecular docking, two basic steps are involved. Okay, two basic steps are involved in docking method. First one is use of sampling method to predict ligand conformation. Okay, so sampling method means first we have to take a sample ligand and we have to dock against the target molecule. So like that, one by one, we have to based on the prediction only, we have to choose the ligand conformation and we have to dock against the target molecule. Okay, so use a sampling method to predict ligand conformation as well as the pose, means the position and orientation of the small molecule, that is our ligand, within the binding sites of within the binding sites and scoring schemes for assessment of binding affinity. Okay, so which shape of our ligand molecule, or in which orientation the small molecule of ligand is able to bind properly towards the active site is identified. Okay, so like that, first we need to by uh, using sampling method, we have to predicting the proper ligand conformation which is able to fix properly to the active site of the target protein, and also based on their position and orientation of the ligand molecule, in which position the ligand is properly fixing into the pocket of the uh, target receptor or target protein. So based on its position, based on its orientation, okay, we need to uh, we need to find out the binding sites, and based on that, in which position the binding is happening properly and efficiently. Based on that, the scoring will be given. All that scoring and assessment indicates us the binding affinity of the ligands. So which ligand have more binding score, which ligand shows more binding affinity, those ligands will be chosen for the next step of synthesizing and testing. Okay, so this entire process comes under molecular docking or docking of ligands. Okay, so in the picture, the target protein is identified from the database, and the different ligand molecules also came from ligand database. Okay, so all the list of targets and list of ligands are given by the database itself, computer itself only. From that we have to choose different ligands and different targets based on their orientation, based on their sizes, based on their binding affinities, and they will be docked against each other, and a molecular docking will be done. Okay, so in the most of the cases, positions of the binding site in target molecule is known, which increases the docking efficiency. Means computer itself gives you the uh, efficient positions of the binding sites. Again, the ranking will be given of the positions of the binding sites which properly fixed by the ligands. So based on that, the docking efficiency will be automatically given. However, in the absence of such prior information, binding site prediction program or online servers such as Grid, Pocket, Surfnet, Pass, MMC, Autodog, Glide, Goal, different types of softwares and databases are available, servers are available, which will give different target sites, different ligand sites, which they will also give you the binding score. Based on the scores, you can download those both the target protein molecules and ligand molecules, and the software itself you can able to bind them, and you will get a proper score. Based on the score, you will fix a certain ligand chemical molecules. Okay, from that few ligand chemical molecules you will again choose the proper chemical structures, and after fixing the chemical structure you can go for synthesizing in the laboratory. Okay, so molecular docking is itself is a another branch. Properly we can go for drug designing by solely itself depending on the docking itself. So molecular docking is it completely another portion of drug design. It is also a bigger branch. You can it is also small kind of ocean. So there are, so in order to simplify this entire ligand based drug design, we can say that there are four steps are there in this ligand based drug design. Okay, in this ligand based drug design, four important steps are there. So I am trying to simplify this LBDD. In step 1, ligand based design methods are based on the fact that ligands similar to an active ligand are more likely to be active than random ligands. Okay, from already literature search, for example, we are going to uh, look for new ligands for tuberculosis. So from literature search, whatever already anti-tuberculosis drugs are there, based on those structures of already presented drugs, based on those structures, uh, active ligands are automatically chosen in ligand based drug design instead of random choosing ligands. Okay, from the database, the similar structure containing ligands are automatically chosen in step 1. Okay, so this ligand based design usually starts with a database which containing a set of molecules with known activities interacting with the same receptor. Okay, so in the database already there are a list of molecules are there with known activities which interact with the same receptors. So from the list of molecules, we have to choose the ligands. Choosing of the ligands from the data self is the first step. So for that purpose, there is a domain expert is present to construct the set of molecules from the database. We can't simply choose the list. So there is a domain expert present, which is already experienced in computer aiding drug designing. That expert chooses set of molecules from the database, and this domain expert is also responsible for dividing the set of molecules into training and testing sets if necessary. So in the computer itself, the molecules are tested and trained and tested for their efficacy. So in the step one, ligands are chosen from the database. Okay. In second steps, the ligand based design is molecular modeling. Means the ligand based approaches commonly consider description based on chemistry. Okay, based on chemistry, based on their shape, based on their electrostatic and their interaction points. Okay, means pharmacophore points. The similarity between these ligands will be assessed in the second steps. Okay, so based on the molecular modeling from the database, the similar shaped structures, similar chemistry structures, similar electrostatic structures are identified, and they were assessed in the second step. Okay, mostly the second step based on the pharmacophore of the chemicals, pharmacophore of the ligand molecules. So what is a pharmacophore? Pharmacophore is an explicit geometric hypothesis of the critical features of ligand. Nothing but pharmacophore can be defined as a molecular framework that defines necessary features that an active ligand should possess for a drug biological activity. Means the pharmacophore is the active site on the uh, target molecule. Okay. So based on this pharmacophore, generally molecular modeling done. If you do molecular modeling based on the pharmacophore, that is called pharmacophore modeling. So in second step, we identify the active site or active pharmacophore, and it will be modeled based on their efficacies, based on their chemistry, based on their shapes. Okay. In third step, the ligand step in the ligand based design usually involves setting up the computational model to identify most promising molecule as lead molecule for further experimental investigation. In third step, when a list of molecules come into the picture, list of acceptors or list of receptors and list of ligands. In third step, again further filtration happened, and most promising molecules were chosen as lead molecules, which enter into the further experimental investigation. So for this purpose, in step 3, the quantitative structure activity relationships generally used in the third steps. So based on the quantitative structure activity relationships only, the modeling will be done here. And from these QSR studies, we will identify one lead molecule or one or two lead molecules in step 3 based on quantitative structure activity relationships, which are also called as QSAR studies. The concept of this QSR is based on the fact that the biological properties of a compound can be expressed as functions of its physicochemical parameters. Means quantitative structure activity means in the lead ligand molecule, which structure shows which activity for that ligand is assessed here. Means all the physicochemical parameters based on the structure of the ligand molecules are expressed as functions in this step. Okay, like proper or popular physicochemical parameters of a compound include its solubility, its lipophilicity, its electronic effects, and the ionization. All these parameters of the different structures in ligand molecules are assessed in step 3, and based on that few lead molecules are identified. Okay, so the goal of QSR studies is to predict the activity of new compounds. Okay, so in step 3, lead molecules are finally identified. One or two lead molecules are identified based on QSR studies. And in step 4, it is the last step to identify the lead molecule from the results of the computational model. Okay, so all these QSR studies and all other steps are automatically done by the computational models only. So finally, from in the last step, okay, a particular lead molecule will be identified. Okay, so here in this picture, uh, there is a huge chemical space, chemical database is available. From this database, the domain expert identified few molecules. These few molecules or few ligands will be trained and tested computationally based on molecular modeling. They refine few ligand molecules which are again tested by computational modeling, molecular docking, and it will give proper ranking to the molecules. And by based on QSR studies, few lead molecules or one lead molecule is identified. All this comes under ligand based drug design. So based on huge database of ligands and target molecules, one lead molecule of ligand is identified based on the ligand based drug design. Okay, so this is the complete structure based drug design workflow. Okay, so from the database we will identify the crystals of ligands and crystal structures of proteins are identified. From the database, known activities of ligands were identified. Okay, and they were entered into both structure based drug designing and ligand based drug design. So from both structure based drug design and ligand based drug design, from various steps, finally a lead molecule is identified. This lead molecule will be entered into the synthesis manner, synthesis in laboratory. Once we synthesize in the laboratory, the bioassay means the clinical trials will be done. If one structure is not showing the proper effect, again we have to go for another structure of lead molecule. Again it will be synthesized in laboratory. Again it will be done preclinical studies followed by clinical trials. So like that here, in both SBDD and LBDD, the screening of molecules will be done. Finally, a lead molecule will be identified, which is go for synthesizing followed by the clinical trials. So like that, commercial drug will comes into the market by following all these steps in by computer aided drug design. Okay. So once we got a lead molecule by both SBDD and LBDD. Okay, so lead molecules will be identified. On this, based on these lead molecules, generally drug designing will be done. So this comes under drug design based on leads. Okay, if you see in the flow chart here, okay, so from the database, targets will be identified. So target validation will be done. So from the target validation, assay development will be done. Means different types of uh, list of molecules will be identified. Through that list, high through output screening will be done, where you will get certain lead molecules will be identified. From the list of lead molecules, again the proper lead identification will be done based on QSR studies. From the QSR studies, one or two lead molecules will be identified. Okay, so once we identified the lead molecules, lead optimization will be done. So up once here the step of lead identification will be done, where the drug design will be based on that these leads only. Okay, so drug design based on leads. So the discovery of a new drug presents an enormous scientific challenge and consist essentially in the identification of new molecules or compounds. Ideally, they will become drugs later. So, the act in new ways upon biological targets specific to the disease, requiring new therapeutic approaches, uh, actually involve in the drug designing based on leads here. So, based on this, today the collective contribution of genomics, proteomics, and bioinformatics, these are various branches of identifying different genes, their proteins, their structures, all these different branches of uh, drug designing, which allows for much more rapid and precise discovery of those genes or proteins which involve in the etiology of certain diseases, which involve in the activity for the cure of certain disease. So, like that, over the last 50 years, most of the drugs commercialized were developed around 5,000 known biological targets. All these were entered into the database. The identification of therapeutic targets require knowledge of disease etiology, means disease, how disease forms, how it is forming, how it is showing its effect. All this comes under disease etiology, and the biological system associated with it were identified as therapeutic targets. So all this information is available for us. So from this database, the identification of the target is identification of the therapeutic targets will be done, and once the therapeutic target has been identified, scientists must find one or more leads, such as chemical compounds or molecules that interact with the therapeutic target with high affinity, efficacy, and selectivity. These three are the important properties of lead molecule. Okay, what are them? Lead molecules are chemical compounds or molecules which are identified from the database, which show high affinity, efficacy, and selectivity. Okay, so the lead compound is nothing but which is demonstrating a property likely to be therapeutically useful. That is the first property of a lead molecule. Okay, which is it can able to demonstrate a property likely to be therapeutically useful. So the property of the lead molecule should be therapeutically useful, and the lead molecule should be used as the starting point for drug design and development. Okay, so the first starting point for drug designing and development is the identifying of the lead molecule. Okay, and the lead is found by design. Okay, design done by the molecular modeling or NMR. Based on that, the design of the lead molecule will be identified, or by screening compounds, natural or synthetic. Okay, we have to the lead molecule will be screened, then it is isolated from a natural extract and is responsible for the extract pharmacological activity. The lead molecule will be extracted. Okay, so once we identified the therapeutical activity of the lead molecule, and then we have to go for drug designing and development, then their structures or designs will be found by molecular modeling or NMR technique and other technique. Once it will be done, these lead molecule will be screened, which will be identified whether they are available naturally or not. If not, we have to synthesize them in the laboratory. If they are available naturally, in which from which natural product, we have to isolate it from that natural product, and we have to test it. If it is not available naturally, then that lead molecule should be synthesized in the laboratory. Okay, so this entirely comes under drug design based on lead molecules. So few important properties of lead molecules are: first one is affinity. The lead molecule should be uh, have affinity towards the target molecule. So this affinity refers to the ability of new compound to bind tightly to its target. Okay, so if the ligand molecules are tightly binding to the target, then it has higher affinity. Okay. Second one is called efficacy. Efficacy refers to the ability of a ligand to elicit an effect on its target that can be measured in terms of a biochemical or physiological response. Okay, means once the ligand tightly bind to the receptor, if this ligand showing some effect on the uh, target molecule, then showing its efficacy on the target molecule. For example, uh, after binding to the target molecule, if ligand is changing the active site of the changing the shape of the active site of the target molecule, means it is showing its effect. Okay, so whatever the effect shown by the ligand on the target site after binding, it comes under its efficacy. If showing more effects, it having more efficacy. So this efficacy indicated in terms of biochemical responses or in terms of physiological responses. Okay. Another terminology is called selectivity. Selectivity of the lead molecule. Selectivity refers to the ability of a compound to recognize its target without interacting with other related drug targets. Okay, so selectively the ligand chooses the target molecule based on their interaction, based on their structure, without disturbing other drug targets, without involving other related drug targets. Means selectively it chooses the target molecules, indicates the selectivity of the lead molecules. So the degree of selectivity required for a new ligand is an iterative process, means an important uh, consideration we need to look for. Okay, so the degree of selectivity also should be good for a new lead molecule. So these are the important properties of lead molecules: affinity, efficacy, and selectivity. Okay, so like that, once based on this, once the lead molecules are identified, then the SBDD technique is effective. So once these lead molecules are identified, okay, in structural based data design, refining will be done after identifying the 3D structures to improve binding to protein and active sites, which is a process called lead optimization. Okay, means the next step after identifying the lead ligand molecules, then we have to go for optimizing the lead molecules. So a few lead molecules are finally identified, which having proper structures, proper binding and selectivity and affinity and efficacies. Then after identifying certain lead ligand molecules, then we have to go for optimization of the lead molecules. Optimization is nothing but whatever the lead molecules are identified, first they will be designed, followed by they will be make into the laboratory, they will be tested against uh, different types of microorganisms or animals or patients. Okay, then it is tested, then it is analyzed. So it is a cycle. Okay, design, synthesizing, testing, analyzing. If it is not showing properly, again we have to design another one, again it is synthesized and tested and analyzed. So this entire cycle comes under lead optimization. Okay, so this lead optimization, researchers systematically modify and refine the structures of the lead testing compounds and docking each specific configuration of a drug compound in protein active site, and then testing how well each configuration binding to the site. Okay, so modeling followed by testing to the binding site. And after that, molecule that is targeted as a potential drug has many required attributes. So once the lead molecule is identified, then we have to test for their affinity, efficacy, and selectivity of a compound, which must be bioavailable and chemically and metabolically stable. Okay, so we have to test their bioavailability, and we have to also test their metabolically stability. So bioavailability is nothing but bioavailability of a compound is depending upon the number of factors, which include its solubility and the ability to cross biological barriers in the body, which is including gastrointestinal tract and the blood-brain barrier, by either passive or active diffusion process or plasma protein binding, etc. Okay, means once we take drug molecule in our body, it undergoing different kinds of activities, right? It has to go through the gastrointestinal tract, it has to go through blood brain barriers, it has to go either diffusion or it should be metabolized. So whatever the process it follow, it should be finally reach to the target protein. So from once taking through the mouth till reaching its target moiety, if the ligand lead molecule is able to sustain, okay, it indicating its bioavailability and its efficacy. Okay, so this bioavailability also should be tested. So once this bioavailability is tested, new chemical entities are identified. A list of new chemical entities were identified, okay, which are also called as NCEs. NCEs are new chemical entities that are highly potent and selective in vitro, lack significant in vivo activity because they are unable to reach their sites of action. So, this may be due to a lack of absorption, first pass metabolism in the liver, or permeability limitations such as the blood brain barrier. Okay, so once the drugs are absorbed in the uh, body system, new chemical entities are released, but they may face difficult types of difficulties in vitro in the body. So they may be permeability issues will be there. There may be lack of absorption will be there. So different kinds of uh, factors influences the bioavailability of the ligand molecules. So all these will be tested. Then only the proper structure, the proper size or shape of the chemical entities are identified. From that, the less ionized compounds means the better it is absorbed and more readily it can pass biological membrane barriers. Okay, so from that, a less ionized lead molecule is identified. Okay, many of the physicochemical factors affecting the bioavailability are poorly understood. Means this bioavailability also should be optimized based on their absorption, distribution, metabolism, and excretion (ADME) properties, which are obtained during the step of preclinical trials of the NCA. So once new chemical entities are identified, their ADME properties also should be assessed. ADME, which we also studied in previous semester, right? Absorption, distribution, metabolism, and excretion. How well it is absorbing, how well it is distributing in the body, how well it is metabolized, and what are the excretion, uh, what are the side effects are there, what are the side compounds are excreting. So all this will be assessed based on that, which is properly doing all these kind of activities will be identified. So one lead molecule will be identified and tested their drug efficacy, and the plasma concentration of either parent compound or its active metabolites also will be identified. So after doing ADME studies, okay, again it will be go for testing. Okay, so design followed by synthesizing and testing their ADME and bioavailability tests, and they will be analyzed in the body itself whether it is bioavailability is more or its efficacy is more, okay, its metabolism is good or not. All this will be analyzed in the body itself. Okay, so like this, all these steps are there in the lead optimization cycle. Okay, so before testing the candidate molecule on humans in clinical trials, scientists must show that the candidate do not present an unacceptable level of risk. given the expected therapeutic benefit. Even though the molecule is beneficial uh, in order to cure the disease, the patient should not be experience the risk. Okay, so for that, all these tests should be done in the preclinical trials. Once the compound has evolved to lead status, it is safety has to be evaluated in suitable in vitro and in vivo models. Then a compound is initially evaluated if there is any mutagenicity or any mutations are happening in vitro. Uh, all this will be evaluated. Okay, it almost takes more than 2 years, okay, in order to show their effects, their reproductive effects, their teratogenicity, and their immunological effects and behavioral toxicity, both in adults and their offspring or their next generation. All this will be tested over more than 2 years. In this way we evaluate toxicological safety of a lead compound. Okay, see for example, recently we experienced corona, right? So in this picture, how corona vaccine or corona lead molecule, drug molecule is identified. Okay, so when once this corona virus showing the effect, okay, the protein related to this corona disease is identified. Okay, so based on the structures of protein which is responsible for corona disease, okay, the proteins are identified by the from the database. From the database, based on molecular modeling, different drug ligand molecules are identified. Again, both ligand molecules and proteins were docked against each other through molecular modeling, and several lead drug molecules were screened and their ranking will be given. Okay, so from the ranking, okay, a few lead molecules were chosen based on their efficacy, rate of binding efficiency, and all other properties. So from that, few lead molecules were identified and they were synthesized in the laboratory. Once these lead molecules were synthesized, they were tested against uh, different types of uh, biological species like rats and other things. Okay, so in clinical trials, once they tested, they were analyzed whether it's showing effect or not. Any side effects are there, whether it is acting properly or not. Okay, they will be done in the analytical phase. Once if they're showing proper effect, okay, they will be chosen as a lead drug molecule. If they are not showing any proper effect, again by doing structure activity relationship, which fragment in the drug is showing poor binding, which fragment of the drug is not binding properly, they will be identified and they will be fragmented, or new functional group may be introduced. All this will be done through structure activity relationship. Again, new structure will be identified if the drug lead molecule is not showing proper activity. Again, it is this designed, so it is a cycle will be done. So for this entire process of lead optimization cycle, it takes more than 2 years. So that's why when this corona vaccine is synthesized, okay, it is quickly introduced into the market before evaluating properly. So that's why people were panicked at that time, because in order to analyze the efficiency of the vaccine, it will take more than 2 years. But within 1 year only, the vaccine entered into the market because of the time limit for in order to take for the lead optimization here. Okay, so generally, the lead optimizations are the general steps of new drugs designed here. Like a chemical moiety structure is identified, then the lead optimization will be done. If the lead optimization is success, then the drug will be synthesized and enter into the market. If the lead optimization fails, based on the various properties such as insolubility, uh, or low specificity, or toxic effects, okay, or toxicity, or false positivity in vitro test, or inefficiency, again lead optimization will be done again, and new structures will be identified. So like that, it will be done in a cyclization manner. This entire cycle of identifying new drug molecules, new lead drug molecules comes under lead optimization. Okay, so lead optimization is very much important in the exam point of view also. Okay, so like this is a general steps involved in designing of drugs, where first basic research into the disease process and its causes will be done, followed by assessment of the biochemical and biological processes of the disease and its causes will be done. Then the team decides where intervention is most likely to bring about the desired results will be done. After that, team decides the structure of suitable lead compound. Okay, so once the suitable lead compounds are identified, then design of the synthetic pathway to produce the lead compounds in the laboratory will be done. Once that will be done, synthesis of its analogs and followed by biological and toxicological testing will be done. All this will be done in the lead optimization process and also in drug designing. Okay, so after lead optimization, then structure activity relationships will be done here. Okay, these structure activity relationships only decides which structure or which analog of the drug molecule is able to showing the proper effect. Okay, and the target molecule is identified. Okay, uh, so this structure activity relationships uh, and based on QSR approach in drug designing, all this will be discussed in next class, because again the structure activity relationships itself is a another huge topic where you need to understand properly. Till now, till lead optimization, if you have any doubt you can ask, where if you are unable to understand properly, you can ask if you have any doubt. చెప్పమ్మా మనోహర్. Of course, yeah, based on guidelines only. Yeah, based on guidelines only means the efficacy rules will be there, the ADME rules will be there. For everything, there are certain rules will be there. These rules are different for different kinds of countries, but mostly they should be followed. Like FDA rules will be there, like different kinds of rules will be there. They have to follow the rules. For everything, there is a rule book is there. ఇంకా ఉన్నాయి. దెన్ మీన్స్ ఆల్ దీస్ ఆర్ కమ్స్ అండర్ స్ట్రక్చర్ బేస్డ్ ఓన్లీ. ఓకే, అండర్ స్ట్రక్చర్ బేస్డ్ డ్రగ్ డిజైన్ ఓన్లీ విల్ బి దేర్, బట్ మోస్ట్లీ the more broader and more important are ligand based drug design and receptor based drug design. Okay, other than this there are homology modeling will be there, but again these are also part of ligand based drug design only. Okay. Hmm. ఇంకెవరు ఆస్య చెప్పమ్మా. ఆసియా షేక్, యూ హ్యావ్ ఎనీ డౌట్? Okay, so once we complete this entire unit, okay, I will share the recording and link with you. Okay, if it is properly recorded or not, I don't know. So I am ending the recording.